Oh. Okay, so thank you everybody for coming. Thank you very much for your questions. They were very interesting and I think I learned a lot answering them. So I'm going to start with a Q&A of this paper, which is the melanocortin action is biased toward protection from weight loss in mice. So the first thing I have to explain is that I made a mistake. So my mistake was mainly that I don't know, because Ilya asked me where did I get this information. I actually read it in another paper, but probably it was like I read it wrong from another paper. So the thing is that the leptin, that I said that activates AGRP neurons, but it doesn't. What it does is actually inhibits them. And the leptin, what it activates are the POMC neurons. So that's the first mistake I made. And the reason why the bidirectional sense of this pathway is questioned is because there are cases of obesity with hyperleptinemia. That's the reason. That's another mistake I made. So I just wanted to clarify that because we're going to talk about leptin later today, or just soon. So that was a thing.
Excuse me. So just for clarification,
Yeah.
leptin also represents basically you can measure how much fat do you have by the amount of fat, because this can be dynamic. Yeah. So at some point it could be really inadequate. So that, as you say, like you have hyperleptinemia, but you still continue to have your obesity, or it is even aggravated. But generally, like the more fat mass do you have, you usually produce more leptin.
Because
so
it's produced by the
this
fat.
is exactly, not only by fat. There are some reports it is also produced by some other cells. For example, it can be produced by the tissues, like the peripheral nervous tissue, in order to promote reparation and healing of neurons. There's also something which is like quite new. I think this report actually had not yet been published, but so that was very interesting report recently in this recent obesity conference in the Cell Symposium just several days ago, and but the major source is of course fat, and this is how, like when you're lean, so you produce like not much of leptin. When you're fed, you basically produce more leptin because you get a little bit more of the fat cells. So it is quite dynamic, and if you're very fat, so you produce more leptin, but sometimes it's not being properly sensed. So this is what is called this leptin resistance, which they also say
exactly.
that whether it is really
like
real
a resistance
or not.
Yeah. That makes sense. Makes sense. If the fatter you are, the more leptin you produce, then might not be a resistant actually.
Right. And there is also like an image within the answer to the reviewers for this paper.
Oh
if
yeah,
you check that.
No, I didn't see that.
So like there are several PDF documents, including the supplement, as well as the answers to the reviewers' questions. So you can really see what the reviewers asked and how they answered.
That's nice.
And they made also a scheme. So they show a scheme where leptin acts via this STAT3, and there it actually also basically converges with the insulin pathway, like both leptin and insulin, to via inhibition of FOXO to also FOXO would then inhibit this action of the STAT3 on the expression of some of the peptides, something like POMC peptide, as well as to inhibit the expression of AGRP in different neurons, of course. But that was the kind of the scheme, like the conventional view. And they say, like in their paper, they are trying to challenge this view. By the way, there is some problem. Is it on NASA's computer?
But allocate. All right.
Okay, I think we can continue now.
Yeah. All right. So, what else? Then you point out this. You said PVN constitutes one of the principal output motor system of the hypothalamus. What I meant, and you asked me for clarification, what I meant is that the PVN is viewed as the nucleus in which afferent inputs from other regions are translated into changes of a specific outputs. That's what I meant.
Yeah, but it's not called motor.
Yeah.
Okay.
Yeah, that was a mistake.
Okay.
All right. So now I start with the questions. I didn't answer in any particular order, just on topics that were somehow related. So the first question is, are there any contradictory findings regarding the bidirectional nature of its action on body weight regulation and the complicities associated with leptin resistant? Is this pathway in clinical practice to be targeted to treat obesity? Do you have any idea about this? And then I reply as well with, how do the conflicting observations impact on the feasibility and rationale of targeting this pathway for developing anti-obesity therapeutics? So I think that also answers a little bit. It can be answered with what Ilya was saying, that the fatter you are, the more adipose tissue you have, therefore the more leptin you produce. So I add that leptin secretion increase on fat state, which in turn inhibits AGRP neurons and therefore reducing food intake. So it has been found that obesity and hyperleptinemia can coexist, and it's hypothesized that it can be caused by leptin resistance. But we don't know if actually this is a leptin resistance. But the point is that increasing leptin secretion, it might not be an efficient method to prevent or reduce obesity, because there are cases of people that are obese and yet they have hyperleptinemia. However, there are some drugs that target the leptin pathway. For example, the leptin have been demonstrated to interact with glucagon-like peptide 1 and its receptor to induce satiation. So GLP-1 receptor agonists are a way to reduce body weight by promoting leptin action. If the glucagon receptor are occupied with an agonist, there is more soluble leptin in the PBH to induce satiety. And for example, a very famous drug is Ozempic, which are the drug that all the celebrities are taking nowadays, apart from some diabetic people. And there are other versions of it that they are also LGP-1 agonists. And for example, a person who takes this type of drugs without being diabetic is Elon Musk, that he publicly said. So, okay, the next question. Is alpha MSH only a natural agonist of MC4R? What is its alternative if there is an insufficient production of alpha MSH? So yes, it's a natural agonist. I don't think it's the only one. And then one alternative is this drug called setmelanotide, which is a medication that acts on the MC4 receptors for the treatment of severe obesity caused by genetic disorders. Again, these diseases include deficiency in the POMC, or in the PCAK1 or in the leptin receptor. But again, these type of drugs, as I mentioned on the presentation, these type of drugs only work when the proteins or the receptors the proteins that are mutated are from the melanocortin pathway. So yeah, there is an alternative. Next question. You mentioned that, any comments? Okay. You mentioned that POMC neurons release alpha MSH that activate MC4R. Does AGRP neurons directly inhibit MC4R, or they also release an inhibitory hormone? So AGRP neurons release AGRP, which is an endogenous melanocortin 4 receptor inverse agonist. These neurons also express inhibitory neurotransmitters such as neuropeptide Y and GABA. So they use different neurotransmitters to inhibit the MC4R neurons. Lastly, will you please explain again the graph I in figure 1? So this is the graph 1, and what this graph measures is the VO2. VO2 is the volume of O2 consumption from the mice. So they do it because the mice, in this case, they have to be on a special cage, which I presume it would be a metabolic cage. There are some metabolic cages that they can measure the O2 consumption, or some others cannot. In this case, they might be using one that does. And then they compare of the two groups. The KIR2 one are these mice that, because we have a supply with the adeno-associated virus, have expressed this KIR2. This KIR2 what it does is to is a potassium is a potassium channel, so induce the inhibition of the neurons. And they wanted to see if the volume of O2 will vary when they express this KIR1 in, I think it was in the POMC neurons. And this the M-cherry is those mice that the only thing that they are injected with is with a plasmid that express M-cherry. The reason why they use M-cherry as control is because we need a way to sort out the cells. How, for example, from the brain, how could you know which are the POMC neurons? How could you sort them with a machine? Like you cannot sort them by hand, and with a incision, like cutting them from the brain, you will take many other neurons that have nothing to do. So we use a FACS sorting, which is a machine that can analyze the cells, can sort the cells one by one, and if those cells express fluorescence, then you can set the machine to say, okay, just collect those cells that have fluorescence. So that's the reason. I think the confusion or this question came because of the M-cherry perhaps, so that's what I'm explaining. So this will be the control.
No, I think that was just the question about the method of what is that exactly.
And then the control and the experiment, and they see no significant difference.
So I think what they used, what I would ask here is do they use here 24 hours average? Because it's milliliter per kg per hour. So is it average of the whole 24 hours, or it is, which I assume it is, because they don't show anything else, because you can also do it separately, daily oxygen consumption or nightly, in the night you are more active, at least mice are more active, so you have to have more consumptions during the night, and maybe there could be some differences during the night but not during the day, or vice versa. So here they don't show anything like that. They show only one level.
Do you think it could be per week, in the 8 weeks, or they wouldn't do that?
That's too expensive. Those cages are pretty expensive. Usually you just do it several days in a row, 3-4 days in a row, and then you just make an average of several days, so that it is per 24 hours. So I assume what we see here is just per hour, but it's kind of averaged they measured all 24 hours and made an average of that. That's what I assume. But in order to see what exactly it is, you need to see the figure legend or probably the methods. I have a question about the previous one. Sorry, I just popped up. So the question was.
Does
AGRP directly inhibit MC4 receptor? Will they release any inhibitor? Oh yeah, okay. So, and you said it is
with neurotransmitters.
With neurotransmitters, but you didn't inverse agonist. Yeah. So you know what is an inverse agonist?
That inhibits. It's an agonist, but doesn't activate the receptor.
Not quite like that. But basically, pharmacodynamically, it is an agonist. So it can activate, but it activates very. It's very poor agonist. So basically, when it binds and kind of activates, it's the same as it inhibits, because there is also some supply of MSH, alpha MSH. And
the receptor is occupied. So.
It is kind of so poor that it basically it is acting as an antagonist. I might be mistaken about that one, but I think that is the way how it works. That's how this classical kind of interaction of those kind of peptide works, and also it is working via because it's inhibitory. It also it also releases GABA, which is also inhibitory. So that's another way. Also neuropeptide Y, but it is your neuropeptide Y is related to the neuropeptide Y receptor, so it is not related to the melanocortin receptor actually. So this is a different pathway. it is also orexigenic, but it is not related to this MC4 receptor. So in this way, like and GABA as well.
It is.
Also not.
Okay.
It also works by the GABA receptor. So it can inhibit those neurons, GABA as well as the GRP, but by different pathways.
Okay.
And also within the arcuate nucleus, there is even interaction of those neurons already there. So that's even before it goes to the dorsal nuclei. So already there could be inhibitory like interactions of those neurons like upon activation. So that is quite complex. And just as a side note, it is not the first report that questioned that POMC neurons, they are critical for the regulation in this, but not in the other context, and that it is kind of not bidirectional. So there were also previous reports that it might be that this axis of the AGRP and PY is actually more important in this regard than the POMC. Also dependent on the context, and also dependent on different contexts. There were really brilliant studies on that, like they were examining the oxytocin receptor expressing neurons within the arcuate nucleus, also these VGLUT2 neurons, which are also anorexigenic, which were supposed to be also like one of those first order neurons in addition to this POMC as well as AGRP neurons, and some other neurons on top of that. So that kind of intricate interrelations, these were already kind of, there were reports who were showing that before. So, and it is also interesting that these guys, these authors, they nicely demonstrated. Okay, so like what happens if we just tackle only this axis? Yeah, we just disregard all others. We just do only this axis, like both the input part as well as the output part, and see like what happens electrically. What happens if we kind of try to tackle the amount of the peptides, and they just basically they showed what people had already knew before, but they didn't have the direct proof. So they had kind of indirect proof that it points to that there is a lack of those inputs, and that this kind of like reciprocal interrelation in case of like induction, like the gain of function of this axis. I mean the POMC as well as the receptive part is kind of, it's not enough. It probably is context dependent, and this is exactly what they here nicely demonstrate. Yeah, I'm sorry for this long summary.
Okay, next one. It would be great if you can explain us about the CRE-MICE and CRE system. Yeah. So sorry, I overlooked that. I didn't realize perhaps I should have given an explanation about this. So I'm gonna talk about the CRE system, and then I'm gonna talk about the CRE-MICE. Okay, so the CRE system mainly is a system that was found in bacteriophage P1. And it has two parts. One is the CRE recombinase, and the other one is what they call LOXY sequences. So the LOXY sequences are equal to each other, and there are 34 base pair recognition sequences. So they have a palindromic structure, which means that it has a the important thing is that it has a very specific type of a structure, and the CRE recombinase is able to identify this structure, and it recombines those two sequences. So depending on the sense these sequences are located, like if they both look on the same sense or not, we'll have different outputs. So for example, we can have an inversion. In this case, the LOXP sequence, they're looking to contrary sense, and inside, for example, there is a gene sequence. When the CRE recombinase intermediate recombines this LOXP sequences, then it will invert the gene, this gene of interest. We could also have a deletion forever. Well, no, it depends. If these two sequences are the same, when they are recombined, then they look the same, then the Cree can invert again and
forever,
again.
Endless.
That forever.
Yeah. This means it doesn't make sense to construct it this way. The first type which you show that will flip flop forever.
Usually
this is really like bad design. So that is how you should not design your construct, basically.
I mean, it depends.
You can do this, but it doesn't make sense.
Yeah, I mean, and there are also other ways, like for example, they could use two different type of LOXP.
Yes.
And that they're like entwined to each other, like intermittent, like A B, something like that. So that way, when one flips, then it cannot make the other invert again.
Exactly.
That's another system.
That is called flex.
That's the flex.
Or DIO.
DIO.
Okay. Double flux inverted open reading frame. DIO or flex. This is fluxed excision. Flex. So that is the same. It's a synonym.
DIO and flex is the same.
DIO is the same. You can call it flex or you can call it DIO.
Okay. Well, it was just like a general idea.
Yeah.
So then if the sequences they are looking at the same sense, then instead of an inversion, when there is like a recombination, you will have a deletion or like excision. And then it can even recombine between different like DNA strands or DNA segments, so you can do translocation of sequences. And then what they use in this case with these mice is that they supply the flex system. The flex is this flip excision, and the flex contains the LOXP, and then inside it also contains a GFP, because we need a marker to select the cells after, and contains the sequences, the FLOXP sequences, and inside an inverse gene, our gene of interest. That could be, for example, this KR2I or the NACBAC. Those are the genes that we're interested in, and they're inverted. And why is this relevant, or why do we use it that way? Because we are working with POMC mouse or mice. So this POMC mouse, what it has is a Cre recombinase that only expresses under POMC expression. So consequently, the Cre will only be expressed on those cells that have POMC. So because we are delivering the system through a very small tiny injection on the brain, and the most likely, I don't know, but ideally, or the way the system is designed, that you only deliver the virus to that group of cells that express POMC, and then you add another layer of control, that is that the Cre would only be expressed on POMC.
You deliver to the whole arcuate.
Well, to the arcuate, to the arcuate.
They will infect all of the cells. Of course, not like there is certain tropism by the adenosine virus serotypes, but generally they will infect multiple different neurons.
Yeah, of course.
So you need to, in order to make it like a cleaner system, you need to use this.
This Cre, POMC dependent Cre.
Yeah, but why not lock stop locks?
They just do it like this.
Do you know?
Sorry, I'm actually not sure if they did the stop, because at least what I saw online, because I couldn't find they didn't give the exact code of their plasmid. They just said it was a flex.
Yeah, flex
blah, blah,
is
blah.
not lock stop locks.
Exactly.
So that's a different system.
Yeah, so they use the flex. They wrote.
So basically, between just for whoever is was asking about this system. So between the promoter and your gene, you can also include locks, then transcription termination site, and then again locks. So by that you can make a recombination also Cre-dependently. And that is what we usually use for the conditional transgene. So my question is, why didn't they use the lock-stop locks, but they use this flex or DIO system? That is my question.
I don't know, maybe because they knew it would have like maybe they wanted to reduce the efficiency in a sense, so like...
No, it's efficient. It's both is efficient. Both is efficient.
Oh, I'm not sure, because Cree has a limit of 5 kb to invert, and this, for example...
That's... Both is possible. So technically it's possible. And they used... Yeah. They made viral constructs with lock-stop locks before. Yeah. But now nobody does it.
With the stop?
Yeah, never.
Why?
Yeah. You only do transgenes like that, but never you do the viral constructs like that. Why is that? Jiang? Do you know that? Yes. Well, you have promoter and then your gene is kind of inverted. Yeah.
The stop box P stop box, the gene was lost.
No. If you have promoter, then you have locks then transcription termination site, which is stop, then again locks, and then your gene. You won't lose the gene. No. You just lose this fragment after recombination. You only this fragment, this transcription termination site will be lost, and then it will read through. You have promoter. That's what you have in the for example, in this Cas9 transgene. So that's very typical for using in transgene. But why nobody uses it anymore in viruses? Any ideas?
Because maybe they don't want to risk that then the stop is inserted somewhere else in the genome.
No, that would not be inserted. We don't have log sequences in our genome. We don't have
no
this
chance.
risk, or at least low risk. Okay, the answer is there is a read-through very often. Unfortunately, even without Cree, it might just read through and have an expression, especially if you have very strong promoter. And they very often they use very strong promoter like CAG, which is a synthetic promoter, and with a high expression of the virus, with a high expression of the gene, you will have read through, and you have expression of your gene even where you don't want it, even when there is no Cree at all. You don't want this. The problem is that with the flex or DIO system, there is the same problem. So there is occasionally also read through. Why? How can it happen?
Because from the other strand, like from the other side.
From the other side? No. It's not. It cannot. There is no it just flipped. The whole thing is flipped.
Yeah.
You cannot. You have a promoter. There is no promoter on the other side.
Yeah, but
it cannot.
Maybe without promoter.
No, it cannot work without promoter. You also need the termination signal. It is because of the recombination. Actually, recombination within the E. coli before you even package the virus. So you while you're packaging the virus, it is already partially recombined because of this recombination events that happen. So when you make subcloning steps, so even in this in the strains of E. coli which are not having the defective for recombination, you still have some chance. So basically, if there was an experiment, they subtracted the LOX-P sites by some kind of scrambled, and then just make some scrambled sequence there, here, and there, and occasionally they see up to 1 or 3% of this unwanted recombination, which happens without any kind of recombinase. So that is what happens. That's why even for this flex and DIO, you need to test, because sometimes the effectivity, this off-target effect of those, in some constructs is higher than in others. So you need to be especially careful when you have some flex constructs. So you need to basically test that, like what is the rate of this off-targeting, and if your cells also are prone to have this kind of off-target effects, or your tissues, you need to. What is your choice? Like how can you deal with that? Okay, just imagine you inject it to the tissue, and there are your target tissue, for example, the POMC neurons, as well as some other neurons, and you detect experimentally that you have like around 1% of activity also in other cells. What can you do? How can you fix that? Any ideas?
To make it more difficult to recombinate, like split the gene perhaps in two segments like this, like log speed, you know, for log speed distributed.
For log speed, it's not a good idea, because it will it doesn't matter like what it finds. It will just recombine until there is no pair left. Yeah, so that is not a good idea. But is there any other way how can improve this system?
With this, we don't know.
We can reduce the titer of the virus.
So
if you deal with 1%, okay, so you reduce the titer of the virus so that you don't have, with a very high titer, it could be like multiple viruses which inject the same cell, actually. So up to maybe 5-10 viruses entering the cell, and then you will have very high infection rate and so on and so on. But maybe you don't need to go that high, so you can reduce maybe 10 times. Like not having like 10 in the power of 13, but 10 in the power of 12 maybe. But this should be empirically tested, so that is important. Need to think about that. But for sure, not lock-stop locks. Yeah, so that is not used in the viral techniques at all. So that is only used in transgenesis.
Okay.
So
That's interesting.
also, sorry for this side note. This was a little bit lengthy, but I think it was important.
Yeah, I didn't know. That's interesting. Okay.
Thank you very much for explaining this whole scenario of how to generate free mice. It was a very much learning experience for all of us. Okay, go ahead, Alejandra, please.
Okay. Thank you for your comment. So about leptin resistant and leptin deficiency, I think we have already talked about this. So it's I think we can pass this because about the obesity, the fat. Okay, thrifty and drifty genes and obesity. So this is one of the questions that make me learn a lot of things, because when I was reading the paper, I was like, okay, I have to look into this because I really don't know what is the drifty. I more or less I assume, but the thrifty I didn't know. So I'm gonna explain it briefly. So these are theories that were originally made to explain why diabetes 2 genetic factors are so prevalent, especially in some like small populations. So the thrifty theory says that some genes related to obesity were advantageous back in the days, like 300 years ago, because humanity went through a lot of starvation, hypothetically, based on this theory, but have become detrimental in the modern world where we have easy access to food. And the drifty theory, it says that these mutations are actually caused by random selection, that in small populations can lead to a genotype domination. So one of the examples they were explaining, one of the reasons that the drifty theory used against the thrifty theory is that there is actually no proof that 300 years ago, 10,000, 20,000, 100,000 years ago, actually humanity suffered of a lot of starvation. Actually, they hypothesized that probably any type of starvation will be caused when humans, or Homo sapiens, start to organize and to grow vegetables and to do like farming, and so, because in that case, when there will be a bad year for the crops, is when they will suffer of starvation. But before, then they will just eat by the day, or they will just eat they will depend on what they found that week or that date. So it's very unlikely that they will suffer from starvation on that time. And this organized farming this organized farming started like 15,000 years ago. So that's actually not long enough for genes to develop and show a prevalence. So that's I think that's a very strong point. More even than the drifty theory itself, that is whatever caused by random selection, is the fact that the arguments they use against the drifty theory. So yeah, that's the explanation.
So again, I didn't quite get it. So you say caused by random selection in small populations.
So,
what do you mean by that? By the random selection?
Oh, so like for example,
selection should be by some pressure, right? So we have random mutagenesis.
Random mutagenesis, yeah.
But selection should be based on some advantage, basically. So it stays in the population if some particular individual managed to give the progeny, right?
The thing is, because it's random, doesn't mean that it's always 50-50. That's the argument they use. So it could be that one of them have, like, because it's not that detrimental, this type of mutations. That's one of the important points they make. It's not something that it kills you from one day to other, or kills you in a very young age. It's something that it makes it
We're talking about which mutations exactly?
In this case, about diabetes 2 genetics. Some of the mutations related to diabetes 2.
Which promote diabetes?
Yes. Not even diabetes 1, but diabetes type 2.
But what caused them to stay in the population? Because like you say, you talk about selection.
Maybe I use it, caused by random.
I don't understand what does it mean random selection. I mean, random mutagenesis occurs all the time, of course. They remain and they remain in the population if they have some certain advantage.
That's the point that they were arguing. That I cannot do that. That that's the point that they were arguing, that they might not be selected because it's beneficial for the population. It might just be selected randomly. That's their argument, because it's true. Also, we have a lot of genetic variants that do not actually the quantity, the percentage of mutations that are actually beneficial are extremely low, way low than those that are actually detrimental. So what they explain is just random, and that it could happen because, for example, if you have like 10 people in a population, 5 will have the mutation, 5 will not. And then the offspring will be random, but doesn't mean it will be 50-50. It might be 20-70. That's their argument, that it might be random. And then
so basically that it had not yet been selected. So like there is this variation, and there was not enough time and not enough kind of pressure to select for one or another. So basically we have the variability, but whatever we see right now is that there is like ever increasing prevalence of the metabolic diseases around the world. So,
but that doesn't have to be genetic cause.
Sure, but that is what they are discussing here. So they want to claim that there is those genetic causes, that they are randomly occurring, but if they are randomly occurring, why do we have the increase of the prevalence there? So it doesn't really explain nicely like the picture. So this is not quite clear for me. So like, what do they mean by that?
Because everything started when they analyzed the prevalence of these genes associated to diabetes type 2 in a group of Native Americans that were living in a very isolated society. So the first theory was that, like, they was giving one of that explanation, and the drifty theory said that how to say, the same way that some of our genes, they have a high prevalence. Like for example, the type of hemoglobin we have usually like has like, for example, in Europe they has the prevalence, and then in other areas has another prevalence. It might, for example, in Africa they have a lot of problems with the sickle disease and with the red cells and blah blah. It might not be because in Africa they needed to capture the oxygen in a different way, blah blah. It can be just because that region evolved in that path, and then perhaps because it didn't have interference from other areas with a different.
No, that example is quite easy to explain. That was in malaria, right? So they survived better.
Yeah, it's true. But in any case, for this one of the diabetes too, that's what they argue. He uses.
Wait, but that's a different here. So
sorry,
in that case, you have a strong pressure
true.
by malaria.
It's true,
So you
true,
basically
true.
don't survive
Yeah,
until
it's
the
true,
reproductive
true.
age.
Okay, bad example.
That's
Bad
it.
example.
So,
It's true.
no, I mean maybe it's a good example. Maybe those genes are good for something else. Could be that they are kind of in some different context they are beneficial. That's why they were kind of selected. That is my point. I mean, like I don't see the selection. I don't understand what does it mean random selection. So I understand what is like random mutagenesis, but like random selection is kind of something which I don't understand. So that was my question.
Yeah, but like for example, I'm sure there are many genes that there are like a mutation on that gene or on the that only affects a 1% of the population, and that mutation is neither detrimental or beneficial, right?
Sure.
And then so the
But this doesn't mean that they are selected.
Claro, but the majority, the one that the type of mutation that the most people have, the type of sequence that most people have, is that selected or not? I would say yes, if most people have it.
Yeah.
And that could be random compared to that 1% whose mutation is neither beneficial nor detrimental.
Right, but what do you want to say? Like we will always have the some kind of variety, and variety is anyway beneficial because at some point some will be detrimental, but some will be also beneficial, or some could be beneficial in some cases, but detrimental in some others, like for the sickle disease, or some other cases,
So so then perhaps the drift theory is talking about variety. It can be just considered variety.
Okay. Yeah, I had the problem with this random selection, so that's what I was asking. Okay. Any other points here? Other comments, Shania or Zisha, or anyone from ivLab? Okay.
Okay. All right. Majority of mutations in the genes implicated in the melanocortin pathway, such as POMC, MC4R, and blah blah, are associated with human obesity. Do these mutations affect the expression in a specific age group? I mean to say, which age group of human is mainly affected? And then she also asked, as the paper showing, the melanocortin pathway is biased toward promoting obesity that is also supported by human genetics, while having no or limited role in obesity prevention and reversal. Do environmental changes trigger this melanocortin pathway in obesity development either in a positive or negative way? So I try to answer these questions, but actually the thing is that even in obesity, what I found is that the main consensus is that obesity is a very complex disease that has a lot of factors influencing it, like either genetic or environmental.
It's even questionable whether it is a disease per se, or
it depends who you ask.
Just one of the kind of symptoms.
A consequence, right?
Yeah. I don't know whether it is nosologically considered as a disease per se.
It's a syndrome, no?
Could be. I'm not quite sure. Like, what is because like every year, like the World Health Organization, they are kind of having this list of, and I don't know whether it is now considered as a disease or not.
Mm-hmm. So, in regard of regarding mutations, they seem that most of the mutations are inheritable, so they will be they will be passing from on the offspring. And for example, just to mention two of them, the MC4R gene mutation is dominant, and apparently the people with MC4R gene mutations tend to gain weight from very early childhood. It can appear as big bone people, like big robust people. That could also be caused by an increase of growth, disproportionate increase of growth caused by a hyperinsulinemia. And then other mutations, for example, those that affect the leptin, like for example the OB mice, is recessive, and these mutant mice are phenotypically indistinguishable from their unaffected litter mates at birth, but then they gain weight rapidly throughout their lives. So I think that every mutation has to be studied specifically, because some of them appear to be exponential, some other appear to be linear. So I think it depends on the mutation that we are talking about. And again, we cannot state whether the obesity is caused only by genetics or only by environmental, because they are still studying about it. And my opinion, I also think that some of the obesity cases might be caused by even more things that depends when you consider environment. I think they also might be affected by behavior, like your own behavior and your like other people's behavior. Because for example, I think, just for what I see, like I don't really have any scientific proof to support my thought, but I think that there's a lot of obesity that is caused by overeating from early stage. Like, typical grandma that pushes you a lot to eat. Like, I know a lot of people that they were raised that way, and after that they just remain obese all life, no matter what they eat. So I think also behavior and acts can affect that.
Childhood obesity is known to be much more severe, and it basically persists throughout the adulthood, and this is well known. As far as I remember, so this is also related to the profound epigenetic change, as well as the microbiome change that is associated with that, and basically that it just persists, especially if this obesity already started from the childhood.
Yeah, so definitely. It also happens as well with the starvation. Like, they found, actually I read a paper recently that there's a huge peak of diabetic people right now in China, that it could be traced back to those starvation times that happened in the 60s and 70s. And it probably can be because these people, when they were children, they went through a very deep, intensive starvation, and maybe they had epigenetic factors that now are causing diabetes. So I think that's also very relevant when we're talking about diabetes and obesity. And she actually asked, the next question is, MC4R loss of function of mutation cause monogenic obesity in humans, that is mentioned in the paper, but my question is in the case of childhood obesity, especially less than 1 or 2 years, like year-old child. So I didn't even know 1 or 2 year old children could be obese. And so she asked if they're the same genes or pathways. So I think, they're the same. But it's also true that when we talk about like childhood obesity, there are more factors, like for example things as well environmental, like the easy access to high calorie junk food, or the few opportunities for physical activity, or lack of playgrounds, and so apart from the genetic factors heritage from the relatives.
Not only genetic, there are several really brilliant studies showing that even epigenetic factors are being inherited from the parents, and when the parents are subjected to like some of the
stress,
not only stress but also some metabolic challenges, so then the offsprings are also getting this kind of pattern, and it looks like it is inherited, so that is really remarkable. They showed it very convincingly.
That about obesity or in general? I saw it about smoking.
Oh no, it was about the obesity traits, the diabetic
obesity as
traits,
well.
and
Wow.
metabolic traits. Yeah, very convincing data.
Okay, and then I left your question for last. So, do they explain the discrepancy between figure S1BC versus D? And what was the reason of such resting potentials in KIR21 and knockback? Please comment about this because I want to talk about it. So the thing is that Nastia, could you, this link I sent you, could you open it please?
You're recording your screen, right?
Yeah.
And Nasty is sharing the screen. Is she?
Because she's also in the chat.
No, I know, but she's sharing because I didn't realize who is sharing and who is recording. So
I am
was
sharing.
confused.
I am sharing. She
Okay.
needs to share. She
Wow.
needs to share.
That's cool. No, just interesting.
Because I cannot enter the Google
Okay.
here, otherwise it gets.
Uh-huh.
Could you share your screen, Nastia?
Yeah.
Do I have to stop sharing? Maybe.
Yeah, you should.
Start sharing.
Okay.
Let's make sure that everyone can see what we are sharing.
If I can see.
Ah, yeah,
Okay.
right.
It's still downloading.
Okay. Is on the one. I think it's the first page, so you don't need to scroll down. Maybe if you download it.
I'm sharing.
Okay, we are very sorry. We have some technical issues. Please stay good, dear YouTube viewers, as
It's
well
working,
as
okay.
dear friends from and colleagues from the Alliance Lab.
Okay. Can you go down so I can see the legend? Okay. Yeah. This is figure 1. Could you go up? Up.
No,
up.
I think it's better that they see the figure, and you need to.
Yeah. Because you wanna.
I hope you know. You can read the legend from my phone if you wish.
You want me to read the legend? Do I have to?
I don't know. You said you wanted
No.
to
I
see
just wanted
the
to see the legend to make sure it was figure 1.
Like this?
Oh, that is the right figure anyway.
Yeah.
That is
That's
figure I
that's
was
the question.
asking about.
Yeah. So you asked B, C, and D.
Versus D, yes. By the way, do you see they made these bars in a way that those red colored circles they basically they don't show where is the average. It could be anywhere. You see, you cannot see that, yeah? It's so unfortunate. I mean, in adult Illustrator, you can just move them. And just like make sure that you at least you see where is the average.
Oh.
But here they just kind of. But anyway, so that my question is like. You see that they have resting potentials, which are different in like this. These are resting potential in D does not correspond what they show in ABC, right? They have.
But D is not a resting potential. D is this, no? Not this.
Yes, but this is.
This
This
okay.
shows like the resting potential, which is also represented there. Yeah, you know. So it's kind of a representative image. You know.
Because this is minus 60.
Minus 76 and minus 80.
Minus 60.
And here like minus 60, then minus 70. Okay, it's firstly it's not really corresponding. Secondly, kind of why it is why it is like that. Yeah. So why they are
Because
why they
I think the ABC might be the average.
not the
And-
average. That's the representative image, not average. That's just a recording from one single
From one-
cell. They just show, and there are so
Yeah.
many cells that they checked. You see, all those kind of circles. Those are different cells which they check.
Yeah, exactly. Okay, because initially I thought it was like they choose a cell that represents the average, so they can show something
That's
graphical in
how they
ABC.
should do this.
And then on the D, they show you how many cells they actually check, and you can see the distribution of it.
That is true. Yes.
But the average should be the values of ABC.
Not necessary. No.
That's the
but,
fair way, no, to do it. That's what I would expect.
Yeah, that's why you call it a representative. But also, in terms of like functionally, yeah. So with the cur, you inhibit, basically you hyperpolarize. With the neck back, you would depolarize, right? And then so that the resting potential changes that way. It is kind of. But okay, let's discuss the second question, right? So my question was.
Comment on S1 recordings. I like to discuss most about this.
SI.
I.
S1I. Right, S1I, that one. As well as S1C as well. Yeah, so basically they also correspond to the same one.
Yeah, this one I don't really know what it represents, this type of figure, because I'm not very familiar with electrophysiology.
Yeah, but you're working in neuroscience, so you should. Okay, let's look at the figure G.
Yeah, right? The figure.
The figure G, no? The panel G. So what are these? What do they do there?
Maybe we can read the legend.
No, you don't need to. Okay, who can tell me what they do there? What they do there? They're stimulating the neurons by the increment of like +5 picoampere, more +5 picoampere, and so on. And then they see what does it elicit. Like when there is a threshold, which causes what? Do you see on the figure G? What are those kind of? Then there is a resting potential. Then it goes. What is this?
Depolarization.
Depolarization, exactly. And if it reaches the threshold,
it's
those
activated.
yes, activated action potential.
potential.
Exactly. So those are action potentials. Yeah. So and after typical action potential, it goes down, right? Because there is also like there should be repolarization, because if you don't have this repolarization period, your next action potential would not occur. That is important. Yeah. So to have this kind of period before your next action potential, and those kind of incremental increase of the stimulation shows like how active they are, and like how much do they require to elicit those action potentials. So that's what is shown on the bottom, about like all those increments, and what it elicits. And you can see that they are really not responsive in case of the CUR2, yeah, because they are basically hyperpolarized. So they need really strong signal in order to get to this threshold, to get at least some of the action potentials. Only in the case of very strong activation. But in case of knockback, which is actually having like much more depolarized state, you see what happens.
They activate very early.
It activates immediately, but it is it a typical action potential curve?
No.
There is no repolarization whatsoever.
So it just remains active. All
Exactly. It is. It is not. It's kind of. It's also called electrotoxicity. So basically, it is. They are being kind of overactive all the time, and without this repolarization, without this decrease, with the repolarization in between, you won't be effective. You cannot basically function. You cannot elicit any kind of, you cannot release anything like that, because you're not working properly. So even though you have this kind of sodium channel there overexpressed, and you have like depolarization, it doesn't mean that you have overactivated neuron. It's in the end, it will be overactivated in the beginning, but in the end it will be dysfunctional. It won't be as dysfunctional as the CUR2. That is why, for me, that part of the story where they use this kind of overactivation and this depolarization is not fulfilling the purpose or the objective that these researchers tried to make. And I'm really surprised that the reviewers didn't pick that up here. Dear authors of the paper, I truly admire. I mean, it's really nice to show like this kind, the whole axis, yeah, from these melanocortin neurons to the melanocortinoceptive neurons, to show like what is the functionality. But this part of the story, I think, this kind of you didn't manage to really reach what you wanted to achieve. You wanted to achieve the kind of hyperfunction of those neurons, but you didn't achieve that.
Oh my God.
Because they were actually dysfunctional, and probably as dysfunctional as the CUR2 one in the end, because they will be active only in the very beginning, and then in the end it is kind of basically no point. Like these neurons will remain dysfunctional. So that part of the story is, for me, kind of questionable. So
that part of the story makes almost the whole paper questionable. The whole theory, the whole hypothesis.
I wouldn't say like that. I would say that this data overall, for me, they are not new. So still, it's great that someone really thoroughly shows what I already used to think of, like even before. I knew that before. Just I didn't have the proof. Now we have the proof that if you overexpress alpha MSH or POMC, or you make sure that you have more MC4 receptor, well, not necessarily you can achieve by that a lot, because maybe you need a specific challenge, like a challenge which they didn't find in this paper, but maybe that is available, for which this kind of action might be actually beneficial. But if you just don't do anything apart from like using the HIFA diet, it doesn't help. Okay, so this I can believe, and I think it's quite fair, and it is important point. But it could be that there could be some other challenges where this could be beneficial, but this needs to be tested. So I don't claim there must be such challenges, but there could be. Okay, but this particular part of the story where they use this overactivating channel under the strong promoter with a virally derived technique, and so this is too artificial and too basically it's defeating the purpose. So it is not actually allowing them to reach the objective that they wanted. So this is my point here.
But this NACPAC system is widely used.
Sure.
Do you think it is all wrong, or they just overuse it?
I'm not saying
If
no.
the promoter is too strong,
No.
is too strong.
I would say like that. Depending on the purpose of your specific experiment, depending on your objective, you can use it. It could be useful. If you are studying some acute experiments, you want to activate something and to see how does it elicit the effect somewhere else. Yeah, that is something different. Yeah, so that's totally fine. If you are talking about chronic, they are talking the whole paper about chronic inhibition, like loss of function or gain of function of this specific pathway. Okay, this I don't buy, because there is the effect of this neurotoxicity, and this is exactly what you have here. And you don't deal with the system when you're going towards some chronic phenotypes. So for chronic ones, My answer is no. Yeah, for some other purposes, sure, it can be. So it all depends on your goal setting. And so if you adequately use the method, fine. Yeah. So even the method which is totally not fitting here could work pretty well in your other system. So that is my point.
But you think that other researchers have not used this knockback as chronic activation? Maybe they have.
Let's look one by one. I mean,
Yeah,
there is no
exactly.
point in generalizing that. But overall, you just, if you're familiar with physiology and overall like how neurons work, you would immediately understand that something is wrong. Especially they have the same data. I mean, they show the data, and the reviewers just pass it through and just because like whatever they set as a goal in the end of introduction, and then they like go and discuss about that in the discussion and the conclusion, and they put it also in the abstract. They pass it through the reviewers.
It eventually goes down, you know? It eventually
Sure,
polarize.
because the neuron is kind of using up all of its energy. Yeah, this is exhausted completely, and it is dysfunctional.
Yeah.
This won't work. Like try to elicit this action potential again. Okay, maybe in several minutes it would work again. It's not working anymore, and with that way, this neuron it will suffer. It's dysfunctional. So basically, they made the gain of function. But this is not gain of function in the end. So this is a model of dysfunctional neuron, or neurotoxically, basically dysfunctional neuron, which in the end is exhausted of its capabilities, and I doubt it can actually effectively deliver the anoxygenic peptides to the site of the action. So. So that's why like this part of the paper is I would say it's weak, and it is I'm surprised that the Nature Communications, right?
Yeah.
So they didn't pick it here. Yeah. So that is strange. Yeah. Okay. Any other questions or concerns or problems or comments?
No, that was my last question.
Okay. That was the last question.
Yeah, to answer.
Oh, okay. Okay. So maybe you want to make some kind of final conclusion, or maybe someone else also wants to add here some kind of overall perception about how well the whole goal setting was, like the design, the hypothesis, and how well did they address it, and so on.
I mean, I think this paper was very beautifully done because they were checking step by step on a pathway. Also, they tested in vivo, so it's always good. And they had, for example, on last paper, I think the one that Sasha presented, we were criticizing that this type of like omics approach doesn't have a hypothesis. And for example, in this paper, it's a very clear. It could be not, but to me it looks like a very clear hypothesis-driven research. So that's kind of like beautiful to see this type of science happening. And yeah, the way it was done, I think it was very clear, very concise. Of course, now if we have to talk about this knockback chronic effect, then there are certain things that have to be considered. But I mean, actually. I would like to research later, like about this knockback in a chronic state to see what are the consequences of such a high activation. But if it really like exhausts the neuron, then there is a bit of a problem with that part of the research. But overall, I think it was beautiful, and I think also. It made me think a lot, like I don't know if perhaps there are other neurons and other neurotransmitters that perhaps can indeed reduce obesity, or it's just perhaps a natural bottleneck, so we will never have such a disease, or it will be very difficult to have a disease or a mutation that leads to a starvation. So it leads to our own self-starvation because of the lack of appetite. I think perhaps has something to do with that. but I think that's also very translational, so I don't know if I can draw that conclusion from the findings
So
we got.
this means you are a proponent of the thrifty theory. So you're rather in the camp of those thrifty guys, yeah? So you think that evolutionary we could not develop some mutation which could lead us to our death from starvation. That's why we are kind of now, because of the overabundance of the foods, we are now kind of more tending towards
Yeah.
right?
But I'm not that informed, so yeah.
Okay. Xiaoyang, in which camp are you? Oh, excuse me. Are you in the thrifty camp or in the drifty camp? Or Nastya or Zishan or anyone else who would like to say anything else about this?
Right. Thank you very much, Alejandra and Elia, for explaining so many technical things and making us learn different new things from this paper and opening up our own brain towards neuroscience and obesity. Overall, this was nice and interesting study and well presented. I would like to just comment that as you have mentioned somewhere regarding one comment that fetal the neonatal obesity or in the childhood obesity. So you had mentioned that for certain diseases like type 2 diabetes, there is program even starts during fetal development due to epigenetic changes, and that interacts with the maternal food intake and what type of food the mother eats and the abundance of that food, the quantity of that food. So that brings about like epigenetic changes, and then that can translate to later metabolic diseases like type 2 diabetes or obesity. So though this is nice study, but tackling obesity unidirectionally or in the linear fashion, I think it will be a challenge because obesity is a broader phenotype and it has many sub-stratification which could be due to genes, due to environment, due to epigenetic changes, due to gut microbiome influence. So generalizing that results from this study will be of limited use for tackling weight reduction or treatment of obesity because they have addressed a few of the issues related to obesity, and there are a number of other issues, and they, in my opinion, they don't work linearly. They work in a network fashion, influencing several genes. Like they have mentioned that there was a thrifty gene and now the drifty gene. So in thrifty genes, I mean, when the food was less abundant at that time during hunter-gatherer time, so at that time, whatever food was available, the body got the capacity to conserve the resources by storing that food and utilizing that during the need. But now the food is abundant. And even Alejandra mentioned that a recent Chinese study in which in 60s, people who were in at that time with limited food supplies, now they have more metabolic conditions like diabetes, type 2 diabetes, I guess. So that could be, genes are there, but how they are acting in the real situation. And these are mutation as well as there are polymorphism, genetic polymorphism. So the drifty genes are like changes in the allele frequencies and then the polygenic risk score, they overall contribute whether a person will develop a complex trait or not. So I think that was related to Rifty. So I mean, my comment is extending briefly. I just want to say that the study is nice, but the obesity as a phenotype is a very complex phenotype, and it is due to several factors. Addressing it in a linear fashion uh, would be helpful, but in certain cases, not in all cases. So that is one of my comments. And if some of my students have any other comment, they can talk about. So, Humera,
Okay, thank you so much.
So this is though obesity is, as Dr. Fazli Abdi said, that it is a complex phenotype. So the authors tried to find some of the way that how one of the neuron or one of the pathway could play a role in the obesity, but it cannot reverse it or it cannot reverse the weight gain. So we can just put apart to treat some of the diseases, but this is not always the case that we can completely reverse the process or such kind of complex disorders like obesity. So it was good, Alejandra. Thank you so much for the presentation.
So before you close, either Alejandra or Ilya, I would like your comment that Cree mice, these have the mutation or the inversion in one copy of the gene, because there are like two copies, one from father, one from mother, or they are changing both the copies of the gene.
If the mouse that they were researching with have two copies of the gene, of what? Of OBOB?
No, in this paper they used only the flex technique.
For the cure.
They didn't use the knockout. Basically, they didn't they didn't need to do the knockout of both alleles on both chromosomes of mouse, because they introduced a transgene. Basically an episome in the form of AAV, and this episome remained as a plasmid basically within the cell, so that is an additional gene, so there is only one allele. Actually, there are many of those viruses which could have entered the cell, but anyway, so that is not about the knockout technique in this case particular. So that's why it doesn't have anything to do about the alleles within the mouse, because that's a gene which is taken which is just added artificially. So that is regarding that particular approach. But you're right. So in case we are talking about the flox mice, where you want to knock out a gene based on the depending on the Cree availability, then you need to have your mouse which has the LOX P sequences which are flanking the gene, and you need to have them in homozygous state, because if you have them in heterozygous state where you have only one of the genes FLOX but the other one is a wild type, then you will still have like one of the copies available even in the Cree positive cells. Yeah. But if you have homozygous floxed mice for the specific gene, which are flanking, where LOX-P sites are flanking the genes on both alleles, then if you have the Cree in that specific cell, then you will lose this gene only in those cells. So that is, but that is a different technique. Like they didn't use this technique in this particular paper.
But it depends on the disease, because for MC4R mutations with its dominant mutation, so with one allele would be enough. And with OBOB, which is those that have leptin disruption, they need, it's recessive, so they need both alleles.
We're talking about Cree-LOX-P system. OBOBs and DBDB models of mice, they are not having Cree dependent, so that they don't need to do this, because like leptin is
But for example,
mostly-
these POMC mice,
Yeah.
they're POMC in one allele or in both alleles?
Yeah.
Maybe that's the question.
You can keep it in both alleles or in one allele. You don't need
You can
much.
be many copies in one
You
allele.
don't need much Cre.
Yeah, you don't need
you
much
need
Cre. Yeah.
two copies of Cre per cell would be enough. It is so efficient that you can even use it in the neuron, which could even transmit just one copy to another cell. So that's what they use by the enterograde tracing. By you basically inject one by a virus, the Cre recombinase, which is expressed in one neuron, and it occasionally, eventually, can also deliver one molecule of Cre to another cell to which it is projecting, and it should be enough to even make the recombination of the report.
heard this in my life.
I actually taught it in my course of viral techniques, by the way, which you attended. Yes, but that's a specific technique. So that is
as a technique. As a technique.
As a technique.
Okay.
Exactly.
I thought you meant happen naturally.
I mean,
okay.
No, naturally, Cre is
Cre
is only available in bacteriophage. It infects bacteria.
Yeah.
It doesn't infect us.
Yeah,
So everything what you do with Cre is artificial. So basically, we've taken something from the bacteriophage, which is used only in the bacteria, and we repurposed it so that we can use it in biotechniques. So that's why we use them.
Yeah, I don't know if this answered your question, Dr. Allen.
Yes, it was helpful. We will dig out more about this.
And actually, I have a question for Sasha, since she made the question. Like you mentioned about children with obesity at the age of 1 or 2 years, but this is a thing that is actually happening. Is this real?
Obesity.
Okay, is it
Yeah,
really
it's
happening?
okay. It's okay.
Is it really happening that 1 or 2
it's
years
really
children?
happening.
Like, are there children that are obese at the age of 1 or 2 years?
Once they also.
I mean, have you seen any
Yes,
case
yes,
like this?
I have seen. Yes, I have seen.
Yeah. Okay. Like there is research that is focused only on that very early obesity.
Yeah, obesity.
Yes, I think there are some case reports from Pakistan about neonatal obesity.
Yeah.
Like as there is neonatal diabetes,
Yeah.
so some monogenic forms of obesity are present in some Pakistani neonates. And these have been reported in
Oh,
literature.
yeah, actually, I have seen 5 to 6 months baby, which
Means,
have
and
obesity.
she basically collected blood samples from the hospital, where she observed some of the children like around 5 to 6 years old, and she found them obese.
Crazy.
5 to
Months,
6 months.
5 to 6 months. Yeah.
Oh my god. And that kid was fed with was breastfed, or it was eating, because technically 6 months you can only eat milk. 5 months, 6 months.
Milk,
They were breastfeeding, yeah. They were on breastfeeding, yeah.
And he was obese. Oh my god. Wow. Okay, that's interesting. I didn't know. Thanks.
Nastya, Shania, Tisha, any questions or comments? Okay, anybody else would like to comment anything? If no, then I would like to say thank you to Alex and to everyone. So, Dr. Awan, or anyone from your side, anyone else would like to say something?
No, I think they have asked their questions and they have learned a lot from this discussion. It was very useful, fruitful. So thank you very much, Alex and Lydia and all the colleagues from your side. So we are extremely thankful for this opportunity.
The pleasure and honor is ours, and we are also very happy to have this productive discussion. Indeed, it was very interesting. Thank you, Alex. Thank you very much. And we see each other in 2 weeks, right? Because we will have, in 1 week, one of your colleagues is going to upload another presentation, right? And then in 2 weeks we are going to discuss it again, right?
Yes, sure, definitely.
Okay.
Yeah.
Okay,
Thank
then
you.
I wish everyone
Okay, bye.
to have a great day and see you in 2 weeks. Thank you very much.
Same to you.
Thank you. Goodbye.
Thank you.
Bye.
Thank you.
Bye bye.
Okay.